And we're going back to plants with a talk by Wellington Muchero.
Thanks for the invitation to give a talk here. We are going to scale up a little bit in terms of organisms, and as has been mentioned, also scale back a little bit in terms of the presentation time. So I'm gonna talk about a really big organism in a really, really short amount of time. And I hope you forgive me for rushing through the slides, but hopefully it makes sense at the end. So instead of giving you, as suggested by the title, my life story on the cell wall biosynthesis in Populus, I'm going to focus specifically on a phenotype that's the end goal for all the work that we do, and that's going to be sugar release. And the work that I'm going to present today is carried out under the auspices of the Bioenergy Science Center, and actually, which is one of the 3 BRCs. And our mandate is to improve yields of biofuels from cellulosic biomass. And we do this specifically at Oak Ridge National Laboratory in our group on Populus. So there are multiple ways that we go about doing this within the plant group, and one of them has been we have done work on activation tagging. We have done reverse genetics using transgenic lines. And my part in the group is with genetic mapping, where we essentially are correlating genotypic information to phenotype and then trying to identify genomic regions or genes that regulate the phenotypes of interest. And the way I go about this is briefly with two main methods. One, which is QTL mapping and association mapping. And that's just the brief comparison of sort of the complementarity that between the two methods that we can get. But the one point that I really want to highlight here, and which is going to be the focal point of this talk, is within association mapping, where I have underlined the sort of the bullet point LD decays within 6 kb. So this is the non-random association of alleles or alleles with phenotype. And essentially what that phenomena enables us to do, we think we can get down to the causal gene when we get a phenotype of interest and a sufficiently large population. So, and I'm going to illustrate sort of that phenomena and how we apply it in the examples that follow. And so the essential things that I need, at least in my work, is one, you give me a phenotypic data and you give me genotypic data. Then I can work all kinds of magic from there. So you'll see some of my latest work related to that. And so the first thing that we did in terms of sugar release was we wanted to sample the natural variation that exists in Populus for glucose release and xylose release. And so what we did was, we sampled 1,100 trichocappa genotypes all the way from just south of here around Sacramento all the way up to British Columbia in Canada. And so what that gave us, and that's the natural range of Populus, and in that we think we essentially captured the natural diversity that exists. And then out of those, the collections that were made in the wild, we phenotyped them for sugar release. And at the same time, we established each and every one of those genotypes is represented in 4 common gardens that are in California, Oregon, and in BC. So, and we have 3 replicates of each genotype. And what that would allow us to do is verify what's environmental noise and what's genetically driven sugar release phenotype. And so what we saw when we collected the material in the wild is the plots that I'm showing represents the extreme phenotypes. So out of those 1,100 trees, I picked the 50 highest and 50 lowest. And then you can see the range the range in variation that's being covered between glucose and xylose release. But then the question then becomes, we are talking about extremely different environments going from Sacramento all the way to British Columbia. So how much of that is just environmentally driven? Is it really genetic? And so the advantage is we just harvested one of those common gardens in Corvallis, which is the Corvallis Common Garden is highlighted by the blue flag over there. And pretty much what you can see is we are capturing, we are almost out of 300 samples that we phenotyped from that common garden. we are almost close to saturating the diversity that we saw in nature. So this really gives us confidence that we are probably talking about something that is really high genetic basis in terms of its expression. And that's always a really good starting point in terms of trying to do genetic mapping with these phenotypes. And so moving on from phenotyping then to how we collected the genotypic data to be able to make this analysis was 16 genotypes were resequenced here at JGI, and Wendy and Joel did a fabulous job aligning these resequencing data and then extracting SNP codes from them. And out of those genotypes, probably 7.75 million SNPs were identified. And then Steve DeFazio, my colleague, and Gancho Slavov led this initiative with Ranjan Priya and Jerry Tasken also playing a part in it, in down selecting from the 7.75 million to a subset of highly informative loci that we'll be able to use in this association mapping. And one of the techniques that they did was haplotype tagging, which means you only pick one SNP that represents a really big section of the genome. You don't really need to assay multiple positions. You assay one position and it still informs you on a larger contiguous section of the genome. And this is going to also come up again towards the end of my talk, and you'll see the utility and how Steve and Gancho were really clever in terms of designing that. And so in the end, they came up with 34,131 SNPs that were used to design the Illumina Infinium array for Populus for genotyping purposes. And then we applied this resource to the 1,100 genomes, and here I just summarized briefly the statistics of the performance of the SNP. Out of the 34,000 SNPs, we got more than 30,000 SNPs that were successfully assayed. And then really high quality data, 919 genotypes had core rates above 90%, and a good chunk of those SNPs had really low missing data, so highly informative data that we were starting up with. And just on the right side is an illustration of sort of the visualization of this SNP assay, where on extreme ends, the red and blue represent the alternate homozygote, and whatever is in the middle, those are the heterozygous genotypes. So then we go to the association mapping part of it, really straightforward. We used mixed linear models, and of these, we had 689 genotypes that we had really good phenotypic data, glucose release and xylose release. And then beyond that, we do population structure and kinship analysis to sort of exclude some of the false negatives. And then in terms of describing the significance of association, we used an LD-adjusted Bonferroni correction, which is pretty much standard. So when you do the association, this is sort of an output that you are likely to get, where most of the SNPs are not significant. If you can see, everything sort of clusters at the bottom, and then there's the one position that's indicated by the red arrow, where we have really high association with the phenotype. And what you're seeing going at the bottom axis from left to right, that's the position on the chromosome. So you're looking at the physical position of those SNPs, and then whatever the sort of correlation between that SNP and the phenotype. And in this case, on this entire chromosome, we only had that one region that was showing high association with combined glucose and xylose release. And then if we zoom in on that position, you notice there, for each phenotype with the key on the left, red boxes represent combined glucose and xylose release. The green triangle is glucose, and then the green diamond is glucose, and then the magenta triangle is xylose. And what you can see is there are two SNPs that are located really near to each other that are about 163 bases apart that are showing the highest association, both with glucose and xylose release and also with glucose alone. And then the xylose is somewhat pretty low on that range. And so the key question I wanted to put across here is these are 13 SNPs within the same gene. So we have zeroed in from the entire chromosome to this particular gene. And in this particular gene, which happens to be a MIB transcription factor, we have 13 SNPs that were assayed. And then we run the association. Out of those 13 SNPs, only those two are showing really high association with the phenotype. So this is getting to the point where I mentioned initially that we thought with the structure of LD and how it behaves in Populus, we should be able to land within a causal gene without additional noise. If this wasn't the case, then most of these SNPs would still show association, and even SNPs that are outside that gene would also be showing association. But in this case, we landed right within 163 bases, we found the SNPs that have the highest association, and they do map in the same gene. So briefly, after we did the entire mapping effort, 40 SNPs had significant associations with sugar release, and 6 were, interestingly, were within intergenic regions, and 34 mapped within 15 genes. And of the 15 genes, 7 were transcription factors. But then what sort of piqued my interest, and I put out that pie chart, which represents many additional other phenotypes that we've been analyzing. So the key thing that quite an abundance of SNPs in the intergenic regions are showing up in these associations, and I was interested to see sort of what's going on here. Could they be in regulatory elements or not? And sure enough, when we did just a quick analysis, they look like those SNPs in intergenic regions fall within known regulatory motifs. And actually, some of the target genes that would be predicted upstream of those regulatory motifs are actually highly suggestive. As you see at the bottom, one of them is a sugar transporter, and one of them was demonstrated in Arabidopsis to be involved in regulating strength of cellulose microfibrils. So it actually looks like, instead of like we normally think about focusing in on coding regions, ESTs, we may actually be missing quite a chunk of the variation that's regulated with intergenic regions versus non-coding regions. So then the key question then that everybody wants to know is, well, yeah, so this gene is associated with the phenotype, but what is it really doing? You know, how much variation can you get, or how much difference are you getting? And the most definitive answer would be with transgenics, where we look at material that only differ that locus. But in the meantime, we can look at the population level differences in allele-specific phenotypes. So what we do is we take individuals that carry alternate homozygote versions of those SNP, and then we just take the average phenotypic value of those, and we, evaluate how much change in mean values are we getting for the phenotypes. And what we see here is for glucose, we get about a 23% change in sugar release, whereas for combined glucose and xylose release, we get upwards of 28%. So again, like I said, the ultimate validation is going to be biological. So we do have these materials undergoing, material being, these genes are being used to create transgenics at Abogen. And then in the meantime, we can do sort of other molecular stuff that we can start to get to the root of what's going on with these genes, and possibly, allele-specific expression profiles. Well, problem was towards when we got this data, this was towards winter time, and the trees decide to go dormant. Apparently, they need some sleep also to be able to function. So we have had to put that expression assay on hold until they break dormancy. Then we can start doing that. In the meantime, a colleague of mine and this postdoc, Jay Chen and Jim Kuo, had optimized the protoplast transient expression assays for Populus. And this was a really perfect way to start digging into some of what's going on with these regions. And the basic idea being, you pick alternate trees that carry the different versions of your gene of interest, you take out the cDNA, you transfect 3 sets of protoplast, 1 control, 1 with version 1, 1 with version 2, and you see what sort of things are happening. And this is actually pretty cool when you're working with transcription factors, because they regulate all pathways. So you can assay behavior of pathways in the protoplast without having to go into the much more complicated sort of transgenic and the heavy stuff. And what Jay, my molecular biologist friend, she says, well, most of these SNPs that you're giving me are in introns. How do I know the structure of the cDNA doesn't vary from individual to individual? And the first thing I told him was that, have faith in me. I will lead you to the right cDNA. Well, so much for that. He didn't believe me. So we had to go back to the resequencing data at least. And JGI have really done a fantastic job of giving us this resource. And what we do then is we can zero in and look at that SNP in context within the resequencing data. And what you can tell is the gene space on the x-axis, where we have intron, coding region, and then intron. And then each individual bar represents an individual genotype that has been resequenced. And so we are looking at the same region across 6 genotypes. And the tag SNP that would have been in our SNP assay would be the one at the bottom. And you can tell if you pick your individual based on that one SNP, you are likely to pick related individual all the way. And this has been truncated for visual effect, but it actually covers a 4.3 kilobase region. And what we are simply doing here is reconfirming what Steve and Gancho had done when they did the haplotype taking, and then pick one SNP to represent an entire region of the genome, and that's exactly what we saw. So my colleague finally believed me that if you can pick the appropriate cDNA based on one SNP in the intron, although it's nice to have resequencing data where we can validate and verify if there are any sort of genomic structural changes that are invisible to us when we use the SNP array only. So in this case, we clone cDNAs from those two individuals that represent the divergent haplotypes, and then we do the protoplast assay, and this is what we see. Two key informations that we derive from this assay is one, in this case, we are looking at a marker gene, which is a glycosyltransferase, highly specific for hemicellulose biosynthesis. And the one key thing that we already know about that gene is that gene regulates the hemicellulose biosynthetic pathway. And in addition to that, not only that, but the two versions of the SNPs that we picked up actually have different ways of activating, with version A having 2 times activating the glycosyltransferase 2 times as much as version B. So obviously the version that you want would be version A in whatever material that you'll be working. And then actually this would be the basis for the differences in sugar phenotype that we will be seeing. And with that, it's unfortunate I could not list all the participants, but this has been a study that involves many, many people, many, many institutions, and those are some of the ones that are listed there.
Thank you, Wellington. Maybe we have time for only one question. If there's any questions. Okay, thank you again. We'll move forward.